So what would the optimal mRNA look like? Well, we want the activity and duration of expression to be tailored to the application, whether that's chronic or transient. We want it to be possible to produce the RNA with financially attainable costs. The template should be designed for ease of manufacturing, and the immunogenicity or the lack of immunogenicity should be dictated by the application, whether that's a vaccine or a genome editing event or whatever. So synthesis and purification processes should be consistent and enable consistent high-quality compounds. And you want to take into consideration the end game as you're designing your process. Plan ahead early for the scale-up of your mRNA, because you may find that the manufacturing processes that can be used at small scales may not be scalable to the scales you might require for your particular application. So think about those kinds of things well in advance. So this is how, in general, we synthesize messenger RNAs. So we use a bacteriophage polymerase called T7 RNA polymerase. And that T7 polymerase reads a transcription template, and that transcription template dictates the sequence of the mRNA that's going to be produced. In that reaction, you can have standard or modified NTPs, and optionally, you can have some cap analogs to do co-transcriptional capping, and I'll talk about that in a later section. A couple optional pieces are inorganic pyrophosphatase and RNase inhibitor. So during the manufacturing process, the first thing we do is we think about the template design and construction, and then we linearize the template. You do in vitro transcription with the T7 polymerase, and then you may include a DNA step to degrade the DNA template. There's a number of post-transcriptional modifications which are possible. For example, if you have not co-transcriptionally added your cap, you can do that enzymatically. You can 2'-O-methylate the RNA to form what's called a cap1 structure, and we'll talk a bunch more later on about cap structures. And you can add a poly-A tail if it wasn't templated. In general, we're doing a purification of the RNA, and then you can have some optional treatments, for example, a phosphatase treatment to remove residual triphosphates. There's generally in-process assays, and then sterile filtration and final release and fill. So during the research phase, the client is generally providing the selection of the target. At that point, the open reading frame is designed, and we'll talk a little bit more about that later. We'll select the 5' untranslated region upstream of the start codon, 3' untranslated region downstream of the stop codon, and this is the open reading frame or the coding sequence here. So at this point, we'll select whether or not we want to use wild type or modified bases, and this may require some experimentation in your model system. We'll determine the polyadenylation strategy and the appropriate purification method. And as Jim alluded to, these mRNAs are structured with caps and poly-A tails, and the function of these caps and poly-A tails and untranslated regions are to bring together the 5' and the 3' end of the RNA and make a little factory where the ribosome's going around and around and around in a circle making proteins. So this is basically what a minimal plasmid looks like. I just wanted to go over some of the elements of the plasmid. So in general, these are bacterial plasmids with an origin of replication. They'll sometimes have an antibiotic resistance gene. Because people are allergic to ampicillin, like myself, people tend to stay away from that selective marker. It has a 5' untranslated region, the coding region, and it has a 3' untranslated region, and then usually a small poly-A tail. And then you'll also want to have a linearization site because the transcription functions by runoff transcription off the end of the message. Now in terms of adding your poly-A tail, you have a couple different options. So you can encode the poly-A tail within the plasmid. The difficulty with that is that long poly-A tails are not stable in bacteria in general, and so you get recombination events. It's fairly easy to get an 80 base poly-A tail to be stable in bacteria, but shorter tails may have lower activity, and that may be something open for discussion after Jim's talk. This may be acceptable in some applications, but it's the most affordable approach. Alternatively, you can use an enzyme called poly-A polymerase to add the poly-A tail. This can give you much longer poly-A tails. We've seen poly-A tails in the thousands if you really want to push it. But there's a significant cost component to poly-A polymerase, and it gives a distribution of poly-A tail lengths. And so you have to worry then in a manufacturing process about the reproducibility of the poly-A tail length addition. And so in many cases, we've had clients ask us to develop a quantitative or qualitative poly-A tail length assay. And here's just an example of what one kind of looks like. So this is just a bioanalyzer trace of an RNA before the polyadenylation event. Again, it's a distribution of sizes with the maximum. And then after polyadenylation, you see you get a distribution of different poly-A tail lengths, and we can take the peak of that peak and compare it to the peak of the original peak and get an idea of the difference in the distributions of those two populations. There are also some other more accurate methods that we can develop for you too as well if you need something more